Hi everyone, welcome to this session. Here I am talking about the protein folding. Protein folding is a process by which a polypeptide chain folds to become a biologically active protein in its native 3D structure. Protein structure is crucial to its function. Folded proteins are held together by various molecular interactions like thermal stability, disulfide bonds, and hydrophobic interactions. This image shows how an unfolded protein becomes folded. During translation, each protein is synthesized as a linear chain of amino acid or a random coil which does not have a stable 3D structure. The amino acid in the chain eventually interact with each other to form a well-defined folded protein. The correct and selective 3D structure is essential to protein function. Failure to fold properly produces inactive, toxic protein that malfunction and cause a number of diseases. Factors affecting protein folding. Protein folding is a very sensitive process that is influenced by several factors including electric and magnetic field, temperature and pH, natural solvent, space limitation and molecular crowding. These factors influence the ability to fold into their correct functional forms. Extreme temperature affect the stability of protein and cause them to unfold or denature. Although denaturation is not always reversible, some proteins can refold under certain conditions. Some cells contain heat shock proteins or chaperones that protect protein in the cell against heat denaturation. Chaperones help proteins to fold and remain folded under extreme temperatures. They also exist refolded proteins in unfolding and refolding correctly. Hydrophobic collapse is the key driving source for protein folding. It formed by hydrophobic core with polar surface interacting with the solvent. Unfinson's experiment. Unfinson's classic experiment for protein folding was proposed by Christian B. Unfinson in 1960. He studied refolding of protein in ribonuclease A, especially concerning the connection between amino acid sequences and biological functions. It contains 124 amino acid residues with 4 disulfide linkages. In the presence of urea, it's a denaturant, and a beta mercaptoethanol is a reducing agent. Ribonuclease A is denatured and the disulfide bonds are broken. When protein is allowed to renature, the denaturant and reductant is goes off. The protein regain its native conformation and 4 disulfide bonds are correctly paired. When the reductant is removed but denature is still present in the disulfide bonds are formed again in protein. Here, most of the disulfide bonds are formed between incorrect partner. This structure of protein called scrambled protein structure. In this image, the first image, the A image shows how renaturation occur when the denaturant and reductance goes off, and the second image, the B image, show how a scrambled protein structure formed. Next one is incorrect protein folding. Protein folding is a very sensitive process, so misfolded protein may happen. Incorrect protein folding can lead to many human diseases. Alzheimer's disease is an example of neurodegenerative condition caused by protein misfolding. This disease is caused by misfolding of the secondary beta sheets of the fibrillar beta amyloid protein present in brain matter. Cystic fibrosis is a fatal disease caused by misfolding of the cystic fibrosis transmembrane conductance regular protein. Huntington's experiment and Parkinson's disease and several allergies has also been caused by protein misfolding. X-ray crystallography is the technique used for the determining of protein folded structures. Crystallography is one of the most efficient and important method for attempting to decipher the three-dimensional configuration of a folded protein. To be able to conduct X-ray crystallography, the protein under investigation must be located inside a crystal lattice. Electron density clouds with the amplitude of X-ray is read the protein folding pattern. This picture shows how a X-ray crystallography works for the determining of protein folding structure. Next is fluorescence spectroscopy. This technique is also used for determining the protein folding structures. Fluorescence spectroscopy is a highly sensitive method for studying the folding state of protein. 3 amino acid, phenylalanine, tyrosine and tryptophan have intrinsic fluorescence properties, but only TYR and TRP are used experimentally. TYR and TRP have high quantum yields to give good fluorescence signal. This picture shows how protein structure gives good fluorescent signals. Thank you. I hope you enjoy this session.